Okay, here's a question for you. If DNA determines literally everything about our health, why do identical twins so often get different diseases? And for that matter, why can something that happened generations ago still leave a biological mark on us today? Well, in this explainer, we're going to dig into the system of genomic medicine and challenge a core belief that a lot of us have about our DNA, a belief that modern science is telling us we need to completely rethink.
You know, the story we've all been told, right? Our DNA is the blueprint for life. It's this fixed, unchangeable set of instructions that decides who we are, what we'll look like, and what our health is going to be. But if that's really the whole picture,
Then you've got this puzzle. How can you have two people with the exact same genetic code, but one gets, say, type 1 diabetes, and the other one doesn't? This one question, this one little paradox, starts to unravel that whole simple DNA is destiny story and reveals something way more complex and, frankly, way more interesting. Right, so in science, the word for this is discordant. Even though they have the same DNA, a gene that's linked to a disease doesn't automatically switch on and cause that disease in both twins. This is really our first big clue that there's something else going on here, something operating beyond just the raw A's, T's, C's, and G's of our DNA sequence. And that leads us to a really big shift in thinking. That old metaphor, the idea of DNA as this static, unchangeable blueprint, it's just not cutting it anymore. It's incomplete. To really get what's happening, we need a totally new way to think about our genes. So here it is. Instead of a fixed blueprint, I want you to imagine your genome as a huge, dynamic control panel, like in a recording studio. The DNA sequence itself, that provides all the switches, knobs, and sliders. But something else is controlling which switches get flipped on or off and how high the volume is turned up. This control panel model is interactive, it's changeable, and it starts to give us a real explanation for why those identical twins can be so different. The actual science behind this control panel, it's called epigenetics. The prefix epi just means on top of or above. So these are chemical marks and signals that literally sit on top of our DNA. They don't change the genetic code one bit, but they tell our cells how to read that code. And here's the really wild part. These epigenetic instructions can stick around for a long time, and sometimes even get passed down to the next generation. Okay, so if these epigenetic signals are like little sticky notes being written in the margins of our DNA, who's the author? Well, it turns out a huge part of the answer is our environment. Let's look at some really incredible real-world proof. This is just a fantastic illustration of the whole concept. Take a look at these mice. They are all genetically identical, clones. The only difference between them is what their mothers ate while they were pregnant. A diet rich in certain nutrients literally changed the epigenetic signals on one specific gene, called the agouti gene. And the result? You can see it for yourself. Huge differences in fur color, in weight, and in their risk for disease later in life. It's a stunning visual example of the environment directly talking to our genes. And this isn't some weird thing that only happens in mice. We see it in humans, too. During World War II, there was a terrible famine in the Netherlands known as the Dutch Hunger Winter. Decades later, scientists studied the people who were in their mother's womb during that period of extreme starvation, and they found that as adults, these individuals had much higher rates of obesity and heart disease. It turns out their epigenetic patterns, specifically something called DNA methylation, acted like a kind of biological memory, recording that early life stress and influencing their health for their entire lives. So now, let's zoom out a bit. We've been looking at our individual biology, but let's look at the bigger picture, the system of genomic medicine itself. Because as our understanding of genetics gets more and more sophisticated, we're running into a strange paradox. Sometimes, having more data doesn't actually give us more clarity. Just look at this number, 3.5 million. When you get your genome sequenced, it's compared against a standard reference genome. Turns out the average person has about 3.5 million single-letter differences from that reference. That is a truly staggering amount of data for any doctor or scientist to try and make sense of. And here's the direct result of all that data. A huge number of those genetic variants end up in a frustrating category called variants of uncertain significance, or VUS for short. This means your doctor might find a unique variant in your DNA, but nobody knows if it's completely harmless or if it's a ticking time bomb for a disease. So in a weird way, getting more data has actually created this massive new zone of uncertainty. But the problem gets even thornier. That so-called reference genome we're all being compared to, it's not some neutral universal standard. As this quote makes crystal clear,
The gigantic databases we use to interpret our own genes are overwhelmingly built using data from people of European ancestry. So what does that actually mean for a patient? It means that a genetic variant that's perfectly common and harmless in, say, an African population could get flagged as being rare and potentially dangerous, just because it's not well documented in the databases. This can lead to wrong diagnoses and create some really serious health inequities. The system itself has a bias baked right into its data. So where does all this leave us? We've seen that our genes aren't a simple blueprint. We've seen that our environment can literally write on our DNA, and we've seen that the very system we use to read our own genes is full of uncertainty and bias. It's time to put all these pieces together and see the whole picture. If you only remember one thing from this whole explainer, please let it be this. Genetics is probabilistic, not deterministic. Our genes don't seal our fate. They just influence the odds. They might load the dice, but our environment, our choices, and just plain old chance all get a say in how those dice are actually rolled. So this is really how our health comes to be. It's an emergent process. Yeah, it starts with the DNA sequence, the raw instructions. But then, layered on top of that, you have the epigenetic signals, acting like dimmer switches, turning gene activity up or down. Then you have environmental factors, what you eat, your stress levels, which are constantly tweaking those dimmer switches. And finally, our interpretation of all of it gets filtered through these huge institutional systems, with all their built-in blind spots and biases. It's a complex, dynamic system. And that right there, that is the crisis in perception. When we get stuck on that old, simple blueprint metaphor, we make a huge and honestly, a dangerous mistake. We start to look at health inequalities that are actually caused by things like pollution or poverty, and we risk mislabeling them as purely genetic differences, as something inevitable. So this really forces us to ask a much bigger question. If we know that our health emerges from this incredibly complex dance between our biology, our environment, and the institutions we've built, how do we start designing a system of medicine that actually sees the whole person and the whole picture? The question isn't just what's in our code. It's how do we build a smarter and a more equitable system to understand it?